decoding microbial genomes to understand their functional roles in human complex diseases. The importance of the gut microbiome in human health and disease has increasingly been recognized in the past several years. For example, in the population-based Lifeline Deep study, researchers found that inter-individual variations of the gut microbiome can be linked to various human phenotypes, including cardiovascular disease, diabetes, and so on. However, we still know very limited about how the gut microbiome can influence human health and disease. In this review, we first summarized microbiome functionalities that may be important for human health and disease. Next, bioinformatic tools that can be applied to decode microbiome functionalities are introduced. Finally, we also introduced state-of-the-art techniques to validate microbiome functionalities. The first part, important microbiome functionalities. The direct influence of gut microbes on the host can be attributed to the fundamental structures that lead to resistance and virulence, such as bowel, which facilitate the survival and activities of trillions of bacteria in the human gut. Specific microbiome structures can help microbes attach human cells or receptors, which further activates virus signaling pathways. Besides, they may carry antibiotic resistance and virulence factors, as well as virus metabolites and other disease-relevant molecules. The gut microbes are also involved in the biosynthesis and biotransformation of a series of bioactive metabolites that can act as substrates and signaling molecules, contributing to normal human physiological functions or complex diseases. Specific class of microbiota-related metabolic molecules mainly include short-chain fatty acids, amino acids, vitamins, bile acids, toxins, and so on. Finally, the gut microbiome can also influence human health and disease through bidirectional interaction with drugs. On the one hand, antibiotics can kill most of the gut bacteria that play important roles in maintaining the metabolic health of the host via a series of mechanisms. On the other hand, commonly used non-antibiotic drugs can be influenced by the gut microbiome via enzymatic transformations that changes their bioavailability, bioactivity, or toxicity. In the second part, tools to profile microbiome functionalities. In this part, we have divided the section into three types of tools. The first one is taxonomic marketing-based approach. The second one is gene homolog-based approach. For this approach, it is mainly designed for shotgun metagenomic sequencing. And the third part is de novo approach. We highlight AlphaFold2, which is a tool published last year in Nature. This tool utilizes different kinds of DNA sequences and further build the three-dimensional structure of the protein and predict the function based on the protein structure. For the part 3, tools to verify microbiome functionalities. Many gut microbes have been found to be associated with various diseases in human participant-based studies. Although the potential functional role of the gut microbiome can be predicted via various bioinformatic tools, the proposed putative functionalities of the gut microbes of interest lack functional validation. Thus, taking advantage of state-of-the-art techniques such as culturomics, genomic editing, novel models, as well as multiple omics may further strengthen our understanding of their functionalities. And finally, might be helpful for the development of microbiome-based personalized medicine. In summary, in this review, we have summarized the functional role of the gut microbiome. We have also introduced the commonly used tools in decoding microbiome functionalities. Finally, we have introduced the state-of-the-art techniques techniques in validating microbiome functionalities. We hope this review is helpful for other researchers.